Hey guys, we're going to keep talking about evolution today, and we're going to take a look at microevolution, which is caused by violations of the 5 Hardy-Weinberg assumptions. All right, so what are the causes of microevolution? We've got genetic drift, gene flow, mutations, non-random mating, and natural selection. So genetic drift is going to be a change in the gene pool of a small population that's going to happen by chance. There's two types that I want to talk about. The first is called bottleneck effect, and then the second is founder's effect. So what we're looking at here is a population. So we've got generation 1. You can see we've got 2 phenotypes, 3 genotypes. So you've got heterozygotes for red, homozygous dominant for red, and heterozygous for white. By chance, only 5 of the 10 plants leave offspring. And so what happens now is we are going to see a change, you can see down here, in the allele frequency. And then if only 2 of the 10 plants leave offspring, we see a further change. And so now we no longer have any of the recessive alleles in this population. The bottleneck effect is going to be a loss of most of the population by disasters. And then the surviving population may have a very different or slightly different gene pool than the original population. So an example here would be if you had a bottle filled with marbles, and that original population would be represented by all of the marbles in the bottle. Now, when you're trying to dump marbles out of the bottle, because of the bottlenecking, where it gets thinner, you're not going to be able to dump as many of the marbles out. You're going to get fewer out, and so that represents the surviving population after the bottlenecking event, and it's probably going to have a very different gene pool than the original population. So if you look back here, there are way fewer white individuals, and there are no yellows, and so now you have mostly blue individuals in the surviving population. So the result is that you're going to have some alleles that are lost, and other alleles could possibly be overrepresented. But generally you see that genetic variation is lost through a bottlenecking event. This is really important because the reduction of a population size could reduce the gene pool for evolution to work with. So if you reduce the amount of variation within a population, then you are essentially reducing what's in the gene pool, and it's going to change how evolution is able to work. Next, you have founder's effect, and this would be genetic drift in a new colony that separates from a parent population. So an example would be older order Amish. You have a smaller population that separated from the old one, and then that population continued to essentially reproduce within that population, and so you see founder's effect. And so again, the result is going to be reduced genetic variation. You'll have some alleles increase in frequency while others are lost as compared to the parent population. Another good example is the Ashkenazi Jewish population. We've talked about that several times when we've talked about Tay-Sachs. And so again, this is important because it's, very common in islands and other groups that don't interbreed, and so you tend to see loss of variation when groups are not able to interbreed with others. Next, we've got gene flow, and this is going to be movement of genes in and out of a population. And so your examples are immigration, when individuals come into a population, and then emigration, when individuals leave a population. So again, the result is going to be changes in gene frequencies within that population. Oftentimes, immigration is going to bring in new alleles into population, and so they'll actually increase the genetic diversity and variation. You've also got mutations. These can change gene frequencies in small populations. It can provide a source of new alleles for natural selection, and it's often actually lost by genetic drift. Next, we've got non-random mating, and this is going to be the failure to choose mates at random from a population. And so you've got a couple causes for this. You could have inbreeding within the same neighborhood, or like animals in areas nearby each other are going to be more likely to mate with each other. You also have assorted mating, which would be like with like. And the result here is it increases the number of homozygous loci. It does not in itself alter the overall gene frequencies, however. Next, we've got natural selection. So this is differential success in survival and reproduction, and this does shift gene frequencies. When we talk about fitness, we tend to have, this idea of survival of the fittest. The stronger you are, the faster you run, the more likely you are to succeed. But this means something a little bit different when you're talking about Darwinian evolution and natural selection. So fitness is going to be the relative contribution of an individual that they make to the gene pool of the next generation. Okay, so it's the relative contribution an individual makes to the gene pool of the next generation. So how many offspring do you have? How much essentially kind of aligns with your contribution to the gene pool of the next generation. Then relative fitness is when you look at the contribution of one genotype to the next generation as compared to the other genotypes. So that would be comparing the contribution of like homozygous dominant versus heterozygous versus homozygous recessive. We've got to talk a little bit about the rate of selection. So the rate of natural selection differs between dominant and recessive alleles. It also can change based on selective pressure by the environment. So if there's a lot of selective pressure by the environment, like let's say there's a really quick, really dramatic shift in the environment, it could increase the amount of selection pressure. So that's something to think about. Modes of natural selection, we've got stabilizing selection, directional selection, diversifying, and then sexual selection. And so we're going to first talk about stabilizing selection, and this is actually going to be selection towards the average and against the extremes. So a good example here is birth weight in humans. You tend to see babies being born, within that like 6 to 8 pound range. You don't want to have, babies that are super small because low birth weight is not a good thing. But at the other end, you don't want to have really high birth weight either, because then that's going to be much harder on the mother. So you tend to see stabilizing selection here, and so you tend to see birth weight kind of come towards the center in a bell curve. Next, you've got directional selection. So this would be selection toward one extreme. So examples could be running speeds in race animals, the Galapagos finch beak size and food source. With diversifying selection, you've got selection towards both extremes and against the norm. And so that could be bill size in birds. You'll tend to see that if you've got like the really big strong bill, that you can get one food source. If it's a little bit smaller, you can perhaps get another. Diversifying selection, though, can actually split a species into several new species if it continues for a long enough period of time, and it's happening in different populations that don't interbreed. So this is just a diagram showing all of these. So with the original population being up here, we're looking at the phenotypes being fur color. With the first one we talked about, stabilizing selection, that would make the bell curve pinch in towards the center. And so you would see, again, like if it were birth weight, you don't tend to see super low birth weights or super high birth weights. They tend to be right in the middle. With directional selection, this is when you're actually shifting the bell over a little bit. All right, so you're moving it into one direction or the other. And then with diversifying selection, this is when you tend to see the two extremes being selected for more than the center. Okay, so in this case, you can see that we tend to see only two phenotypes of these fur color. And this is the one that, if you continue with diversifying selection for a long period of time, could lead to new species. Sexual mate selection may not actually be adaptive to the environment, but it increases reproductive success of the individual. And this is actually a really important selection type for species. And it results in what's called sexual dimorphism. And so that's the differences in secondary sexual features between males and females. So we have a couple diagrams here. So you've got the big mane on a male lion, or like the large antler rack on a buck. All right, so those are secondary sexual features, and they're used to attract mates. So females often drive sexual selection and dimorphism because generally females are the choosy gender. They're the choosy mate, meaning that they devote so much time and energy to raising and caring for the offspring, that they want to make sure that they're choosing the best mate possible so they have the best offspring possible. And so they're going to be the ones driving this sexual dimorphism, because oftentimes, like if you're talking about deer, the male with the biggest antler rack would be the most successful. And so females are going to tend to choose those types of animals to mate with. Ways of preserving genetic variation. One is diploidy, which is preserves recessives as heterozygotes, then balanced polymorphisms. This is a preservation of diversity by natural selection. So an example would be the heterozygote advantage, and this is when the heterozygote or hybrid survives better than homozygotes. It can also be called hybrid vigor, and the result is that you can't breed true, and so the diversity of the population is maintained. Just to kind of clarify, an example here is sickle cell anemia. And so when you have, when you are homozygous and you don't have any alleles for sickle cell, you are not resistant to malaria. When you are homozygous recessive, you have sickle cell. When you are heterozygous, you have a resistance to malaria and no sickle cell. So that would be considered hybrid vigor. And why this is important is because the allele for sickle cell tends to be really common in many parts of Africa, which is also where you tend to see malaria. And so if you have that heterozygote, so you've got one normal allele, so you don't have sickle cell, but you have one sickle cell allele, you have resistance to malaria. So population geneticists believe that all genes that persist in a population must have had a selective advantage at one time or another. And so again, that's where this sickle cell allele comes in. So you don't want to be homozygous recessive, because then you have sickle cell anemia. You don't want to be homozygous dominant, because then you have no resistance to malaria. But if you are a heterozygote, you sort of win at both. So the summary for this chapter: you guys should know the difference between a species and a population. You need to know what the unit of evolution is, that we should be looking at populations and not individuals. You definitely need to know the Hardy-Weinberg equation and how to use them in calculations. And then you also need to know the Hardy-Weinberg assumptions, and then what happens to a population if an assumption is violated. You need to know the various modes of natural selection, so the directional, diversifying, all of that good stuff. And then you need to identify various means to introduce genetic variation into a population. And so your lecture question for today is, does evolution ever result in perfect organisms? When you tell me your answer, make sure you explain why you said yes or why you said no, and please send me your answer to my email via the class website. Thanks, guys.